going. So next up is Qiaoli Li from Thomas Jefferson University as well, and from our center. Li, we've been working with a very long time, and you saw both Lili and Kuhn in the video that we played yesterday morning when we were doing our tribute to Yoni. So Lili, turning it over to you.
Thank you, Sharon. My presentation deals with functional assessment of human ABCC6 missense variants. Missense variants are single base pair substitutions at the DNA level, which alters the genetic code, producing a different amino acid at that position. So for example, here at the DNA level, there is a nucleotide A, which becomes C, and that causes a change in the amino acid. So my work deals with a type of missense variant, which are the most frequent in the human ABCC6 gene. Annotation of this type of variants have primarily relied on predictions by in silico algorithms. For instance, the American College of Medical Genetics and Genomics set up guidelines as to classification of missense variants. And here, there are 5 different groups. So if you have contributed a blood sample, a saliva sample, or buccal smear for mutation analysis, and you received a report of your genetic testing, you may find those classifications in your report. So from the end, if the sequence variation may be a benign variant, likely benign, which do not cause PXE. And there are some variants are classified as VUS, variant of uncertain significance, meaning that the in silico predictions do not really annotate this variant as pathogenic or benign. So there's lots of uncertainty here for those type of sequence variations. And there are also variants classified as likely pathogenic or pathogenic, and probably contributing to the diagnosis of PXE. So with this kind of genetic predictions, we take a further step here in the lab to not only resolve those variants that are classified as VUS, but also for those pathogenic or less than pathogenic mutations, how do they exactly contribute to the disease? Because ABCC6 is a transmembrane transporter and it is permanently expressed in the liver. So can we experimentally analyze this type of variants in an appropriate model system? So the ideal model here to do this type of analysis is to obtain a liver biopsy from patients for us to look at what went wrong in the ABCC6 protein in a patient liver, but that is not possible. So my laboratory took an approach that we can analyze these variants in the PXE mice. So for instance, we wanted to look at 3 different missense variants present in the patient's DNA. And this variant is classified as VUS, variants with uncertain significance. And that there are two other variants classified as pathogenic. Our approach is a adenovirus-mediated liver-specific human ABCC6 expression in ABCC6 knockout mice. I give credit of this PXE mouse model to Uni Vito, MD, PhD. This model was established before I joined this lab. And this mouse model is very useful to study not only the pathomechanism, but also, as you can see in this presentation, as a model for gene therapy and looking at the different ABCC6 missense variants. So what we do here, we took those mice and we inject those mice by tailwind with an adenovirus expressing human ABCC6, either wild type or mutant. We can introduce those variants one at a time. And then we inject them side by side with the wild type human ABCC6 virus. And we inject the virus at the same dose, by the same injection route. And then we wait for 4 weeks. 4 weeks later, we euthanize the mice. We collect their livers to look at whether the wild type or the mutant human ABCC6 protein is expressed. If it is expressed, where it is localized, because the localization of the protein in the liver is very critical for ATP release. And then we also collect the blood from the mice. We measure an important molecule in PXE model. You have heard from other people's work, that is inorganic paraphosphate. The wild type of protein would contribute paraphosphate to the blood, and now be asking whether the mutant protein would do the same. And we also collect the muscle biopsies from the mice here, because these are the specialized hair follicles called vibrissae that becomes mineralized. So if the human protein is functional, and the same protein in different species do the same thing, and the wild type protein would rescue this calcification phenotype. And a mutant and a non-functional protein would not rescue it. So this is the approach that we take to analyze those variants one at a time. So they are our wild type and knockout controls. So they don't express the human ABCC6 protein. And if we do a special staining called Fancasa for the muscle skin containing vibrissae, and you see here those black mineral deposits in the vibrissae only in the knockout mice. And we measure paraphosphate in the blood. The paraphosphate in the blood of the knockout mice are low, causing them to calcify in the muscle skin biopsies. If we inject those mice with wild type human ABCC6 adenovirus, and if we look at their liver, look at this beautiful green fluorescence in hepatocytes of the liver. So this is a beautiful expression of the human wild type protein. And if we measure paraphosphate in the blood, okay, that brings the paraphosphate up to the level of the wild type mice. And that prevents calcification in the muscle skin. If you look at the histology here, there's no calcification at all. So the human ABCC6 protein is an ortholog of the mouse protein, and they do the same thing. So based on those results, let's look at the first missense variant. So this is the leucine becomes valine at amino acid 420 in the human ABCC6 protein. So is this mutant protein expressed in the liver? Yes, it is expressed in the hepatocytes, on the cell membrane, as the same as the wild type protein. Does it increase paraphosphate in the blood? Yes, this is very similar to the wild type protein. And because it drives paraphosphate up to normal level, those mice do not calcify in the vibrissae. And they have very low levels of calcium hydroxyapatite in the muscle skin biopsies. So this variant, classified as VUS by ACMG, is now confirmed to be a benign variant. It does not cause PXE to happen in the mouse model. What happened to this variant? G becomes R at amino acid 1302. The protein is also expressed at similar levels to the wild type protein on the cell membrane. But when we measure paraphosphate in the blood, the paraphosphate level is low, is very low, which is similar to the knockout mice. And because paraphosphate is low, there's so much calcification in the vibrissae. So this variant, classified as pathogenic by predictions, is now confirmed to be indeed a pathogenic variant. But these systems allows us to understand what happens, what exactly happens, how does this pathogenic mutation contribute to the PXE phenotype? And the defect of the mutation is not with protein expression, is not with the cellular localization of the protein, but it is not able to mediate ATP release from the liver to the blood circulation. This is the last variant. Arginine becomes W at another different amino acid position. So look at the liver of this mouse. So the protein is expressed at a very low level. And interestingly, this protein is expressed intracellularly. not on the cell membrane. You know, ABCC6 has to be on the basal solid plasma membrane of the hepatocytes to, you know, transport ATP from the liver cells to the blood circulation. So when we measure pyrophosphate, and yes, indeed, pyrophosphate in the blood is very low. And those mice develop extensive calcification in the rebrissae. So this variant is also a pathogenic mutation contributing to the PXE phenotype, but the defect of this mutation is expression and cellular localization of the protein, making the mutant protein not functional. Utilizing this functional system, we analyzed a total of 10 human ABCC6 missense variants that we found in patients with PXE. So you can see here, we follow the same approach. We look at expression in the mouse, cellular localization, we measure pyrophosphate and the calcification phenotype. And our experimental data, in some cases, are different from in silico predictions. So just to summarize, so we think that the PXE mouse model is very useful to elucidate the functional consequences of human ABCC6 missense variants. The studies, however, have limitations. As you heard from Olivier, the mice are not human. So here we studied a human mutation in mice, and we used the cDNA instead of genomic DNA for the study. And the promoter is driven by CMV promoter, which is not the endogenous ABCC6 promoter in the liver. And therefore, the variant-associated clinical decision-making should be made with caution. So this is the group of people that I have been working with over the years, not only for the genetics work, but also we use the PXE mice to test different compounds, therapies to prevent calcification in those mice. We are very thankful to Sharon Terry and her team at PXE International for her support over the years. And we thank all the patients and the families who contributed to our genetics studies in the past. And this is the photo we took last year with Mary from PXE International. And I truly appreciate the support from Dr. Qiaoli Li. I have been working with him in the same lab space for 17 years. And this is Kuhn working with me in the center. Thank you very much.
Great. Thank you very much, Li, for all your work all these years and for keeping the light burning. Questions for Li? Maybe I'll just say this work is very important in the sense that we, for example, PXE International regularly gets requests from geneticists, from pediatricians, from regular clinicians, and from patients themselves asking, is this report that says I have a variant of unknown significance significant? And we often turn to Li and her team to ask, what do you think? Because the database says one thing, and in fact, it might be another, as you've seen by her work. Also to note that the large database of variants of mutations that is curated now by the National Libraries of Medicine was curated first in Europe and the Netherlands, and PXE International took over the curation of ABCC6 once we knew that it was our gene, and then now works very closely with a particular person at the National Library of Medicine at the National Institutes of Health to get these things corrected when Li or others find there's incorrect information. And we also upload and bank everything that we can find. A couple of years ago with Qiaoli and Li, we looked at 500 of you and your mutations, and were able to discover a whole host of new variants that we were able to put into the databases so that researchers and clinicians all over the world can just look in that one location and find the information. Okay. Oh, a question is, a missense variant different from a deletion in ABCC6? If so, Do we have a sense if there's a difference in the impact of missense versus nonsense versus deletion versus the various types of mutations?
Oh, yes. So this is a very interesting question. So there are different types of mutations. Deletion mutations are different from missense because when the gene has a mutation, so the protein can be truncated while remain in frame, which means your protein may be short of a few amino acids. But sometimes the deletion mutation will throw away the reading frame, which means the protein is truncated and it's short in translation and it does not make a functional mutation. But missense variants are the most frequent type of variants in human ABCC6, while most of the variants are private and running in only a few families. And missense variants work differently in that the full protein will be translated in the full length. It is just there's 1 point amino acid that is changed. So that makes the distinction between a missense variant and a deletion mutation.
Thanks, Lili. Thank you very much.